お時間になりましたので始めましょう。こんにちは日本、Hello Europe, Dobar dan Srbijo. Gde je moja Srbija? I think I see my country. It is very nice to see you again for this webinar, and I'm very, very grateful to Professor Hayashi, who accepted to give us a talk today on a very, very interesting topic. So before we start, I would like to say Hayashi Sensei is really a long-term contributor to aquaphotomic science and generally in water science research, especially in relation to interaction with materials and biomaterials. So, and lately he seems to have very interesting findings, and we hope that we will have some interesting collaboration in future. The professor will give a talk for 1 hour, and then we will have half an hour for discussion. But please, you can actually give all the questions during the seminar. You can just type the questions in the chat box. Webinar will be recorded and available on our YouTube channel soon after today. And after that, at YouTube channel, we will also have subtitle in Japanese. Just one more information before we begin. April, there will be a next webinar on the actually development of Aquaphotomics research department and what we did in the past year. So I think I said everything. If you have any questions, stop in the chat box, and let's now enjoy the lecture by Professor Hayashi. Hayashi Sensei, go ahead. Thank you very much.
Yeah, thank you. Thank you for your introduction. So I will share the screen, just a moment. Okay, can you see my screen?
Yes, yes.
Okay, so I will slowly start. So first of all, thank you, Professor Zenkova and Elena, for giving me a great chance to give a talk here. Yeah, thank you very much. And I'm very excited. And I believe this seminar is a little bit different from the very official one. So in my talk, so I would like to introduce our recent results, and basically it's a behavior of water surfaces. And actually we have a lot of unsolved questions. Then, so maybe I will ask you some hints in a lecture. Yeah, so let's start. So first, let me start with the introduction of our laboratory. So our laboratory is very strong in analysis of the surface and the interface. And I think we have three major topics. So one is instrumentation to observe the interfacial phenomena of molecules at the interface. So especially, so we love scanning probe microscopies, atomic force microscopes, and we use fluorescence and IR Raman microscopes, and sometimes we combine them. And actually, some of them are homemade, very original techniques. And second topic is we always want to collect the information on proteins or cells' response to biomaterials. So to collect the data, we try to make platforms, so which approach to generate a big data. So surface with a chemical gradient. So we use surface and monolayers, lipid bilayers, polymers, and the metals and oxides. And when we get large scale of data, so we will use the techniques of informatics to find the hidden correlation between the chemical structures and the biomolecule and cell responses. And we try to make some strategy to suggest the new materials with desired functions. So that's why I named our labs biointerface and informatics lab. So it depends on main topics, the name. So we change the name of our lab flexibly. So let me introduce us some activities. So today we will mainly talk about the interfacial water, role of interfacial water at the biointerface. So especially the interface between biomolecules and cells with artificial materials. And so we have studied molecular behavior of water at the interface. Recent research. So we try to predict water contact angles. It's a very basic physical-chemical property of materials, and also adsorption of proteins from the chemical structure of the materials. And we are also interested in instrumentation. So one example is visualization of drugs in the cells with a homemade Raman microscope, vibrational spectroscopy with a mapping function. So we can visualize the localization, concentration of drugs in the cells. And we can also visualize the molecular binding site on the biosensors. By using the ligand-receptor interaction, and we combine with the techniques of scanning microscope, scanning probe microscopes, so we can visualize where the molecular binding occur at the nanoscale, so with the nanoscale resolution. But today's topic is the origin of biocompatibility, blood compatibility. So let's go to the main topic. So first, I would like to show you the two scanning electron microscope images. So I brought two materials to the collaborators, so Professor Masaru Tanaka in Kyushu University. And so they have a staff of medical doctor, and he extracted my blood from my body and put on a surface. Then we rinsed with buffer and observed electron scanning microscopes, SEM. And so you can see the left image. There are a lot of something on a material surface. Actually, these are my platelet cells. So platelet cell is a kind of a UFO-like shape, and its size is from 4 to 5 micrometer, but so it's completely flattened. And if you can check carefully, so cells are trying to connect each other. So this is an activation of platelet cells. So in this case, the platelet cells recognized this surface as an enemy and try to cover them with forming aggregation. So actually, so this is a activation of the platelets, and if the material surface induces such kind of activation, so if the aggregation is stuck in our brain or our heart, so we will have a serious problem. On the other hand, so right image, so the material surface is completely stealth against the plate. Actually, these are monolayers with the same molecule, but completely different blood compatibility on the surface. And actually, so for the artificial heart and lung, or recently ECMO, so artificial heart and lung for the serious coronavirus patient. So the blood compatibility is very important material function. So this is my favorite Japanese comic, and the story of the professional doctor, medical doctor. So he's a professional of the surgery operation. And there is a one episode story on blood compatibility. It's a blood clotting inside the artificial operation. But this is just a problem of blood compatibility. So he could not solve this problem. And actually this story was published in 1977, almost 45 years ago. So we had such kind of problems and problems of the blood compatibility, and we have not solved these problems until now. Actually, this is a very important, difficult problem question. So one example is so Professor Buddy Ratner at the University of Washington. So he's a professional of the blood compatibility. He's from the field of surface science. And in 1993, so he published one review article, and the title is the blood compatibility catastrophe. And he mentioned, so blood compatibility is really mysterious. And after that, so 2007, 14 years later, so he's published one review article, so the catastrophe revisited. So in this article, so he also mentioned he could not solve the problem of the blood compatibility. And so 4 years ago, so he published one interview article, and he also confessed that he could not solve the mechanisms of the blood compatibility. Actually, so there are, in general, so if we want to make surfaces bioinert, anti-biofouling, the general way is to cover the solid surfaces with a hydrophilic polymer. So if we immobilize the surfaces with hydrophilic polymers, the polymer chains are free hydrated, and the degree of freedom is very high. So if the biomolecules want to adsorb on the surface, so they need to shove the polymer chains away. So, but you can compare this and this. So this process is really energetically unfavorable from the viewpoint of entropy and enthalpy. And so recently, so we also knew the hydrophobicity or hydrophobicity of the polymer is not so important. So this is this makes the problem very difficult. But fortunately, we have several very good blood compatible polymers. So one is a PMEA developed by the Professor Masaru Tanaka. So when he worked in a Terumo company, and this is a chemical structure of PMEA, and you notice the chemical structure is not so complicated, but it has a very high blood compatibility, and this polymer has a #1 market share for the coating material of ECMO, artificial heart and lung. So the other example is a MPC polymer, so developed by Professor Ishihara from the University of Tokyo, and he mimicked chemical structure of lipid molecules constituting a cell membrane, and so the side chain of the polymer have phosphoryl functional groups. So negative and positive charges coexist. So these polymers have a completely different chemical structures, but they show very high blood compatibility. And now they are still working on mechanisms. Actually, I wanted to work on the polymer systems, but polymer is very complicated, and interface between polymer and the water is not so clear because polymer chains are fluctuating like a seaweed. So first, we wanted to what kind of interaction is responsible for the blood compatibility or protein resistance, cell resistance. So we go back to the very fundamental system. So this is a so-called surface monolayers. So in this case, so I would like to introduce the surface monolayers, SAMs of the alkanthiols. Very stable metal, even in air and water, but very reactive to the sulfur. The sulfur and gold can form a covalent bond. And due to the substrate molecule interaction of the gold sulfur bond and the intermolecular interactions, so the thiol molecules of the gold forms a very highly ordered self-assembly monolayers. And actually, it's very easy to make such kind of monolayers. Just immerse the gold substrate into the solutions containing thiol molecules. Then we can get such kind of molecules, monolayers. So it's easy to make, but the biggest advantage of this monolayer is if we choose the functional groups, so we can control the physical chemical property of the surfaces. For example, if we put the nonpolar at the end of the molecules, then surfaces will be hydrophobic. On the other hand, if we employ the OH terminated or charged groups terminated alkanthiols, then we can make surfaces hydrophilic. So we can change the polarity and the charges of the surfaces just by changing the molecules. So there are several kinds of the nonfouling, anti-biofouling surface monolayers. So they are categorized into two groups. So one is a non-ionic type, So the most general one is oligo-ethylene glycol terminated surface and monolayers. So this is a chemical structure of molecules. So there is a anchoring group and a spacer group. This enhances the van der Waals interaction. And as a terminal group, if we put just 2 or 3 units of ethylene glycol, then the monolayer will be the anti-biofouling. This is a non-ionic type. And for the zwitterionic type, so we have a mixed charge type and zwitterionic type. So if we mix the 2 molecules, so negatively charged sulfuric acid ones and positively charged trimethylamine. So just we mix them, make a monolayer. So such kind of monolayer is very blood compatible and protein and cell resistance. The betaine type, negative charges in a 1 molecule. So such kind of monolayer is also very bioinert. On the other hand, so we have a lot of protein and cell adhering SAMs. So one of them is a hydrophobic methyl-terminated SAMs. The water wettability is very low, non-polar, just a typical non-polar surface and monolayers. And hydrophilic SAMs are also protein and cell adhering. For example, neutral OH terminated, the slightly negatively charged COH terminated, and slightly positively as they absorb proteins and adhere cells. The question is, so what is the difference between these SAMs and previously introduced blood compatible SAMs? So I will introduce the strategy. So our idea is very simple. If the surfaces really reject the protein adsorption or cell adhesion, so such kind of surface may induce a repulsive force to reject them. And if we approach such kind of surfaces each other, then we expected, so there is some repulsive force. And if we can clarify the physical origin of the repulsion, so this will be the mechanism of the bioinertness. So for this work, so we use the atomic force microscope. So atomic force microscope is a tool for visualize the nanostructures just by scanning the surface with a sharp tip. This is a normal use, but in this research, so we try to measure the force between 2 objects, especially, so we fix the silica colloid sensitivity in a force compared with the sharp tip. And we made such kind of system, and we fabricated the surface and monolayers on silica spheres and also on substrate. Then we approached them each other in a physiological condition. So in this measurement, so we get the force and the distance curves. So if we observe the repulsive force, the profile is like this red curve. So if there is an attraction, it shows the force. So if you remember this, so you can understand all of the experimental results. So if it's repulsive, it's a positive. If it's attractive, it's negative. So let's go to the results. So first, we the force in a pure water. So the surface monolayers is Eg3OH. So 3 ethylene glycol units terminated with OH. And we can observe the very long-ranged repulsion on the more than 100 nanometer. And if we increase the ion concentration, so this repulsion completely disappears. So this strongly suggests this is a electrostatic double layer force. And if we use a DLVO theory, which describes the interaction in a estimate the charge density. Actually, this if we calculate the charge density, so the density is a 1 charge per 13 by 13 nanometer square. It's a very low concentration, but it induces a very long-ranged electric double layer force. Which charge, negative or positive? So in this case, so we can just change the surface monolayers to positively charged ones. So the interaction became attractive. So this means the charges on near this monolayer is a negative charge. Actually, this is a hydroxide ions in a pure water, so generated by autoionization of water. But not only the long-range repulsion, so we observe the short-range repulsion. The concentration. So we will discuss this repulsion. So before that, so I just want to compare the force profiles for the various SAMs. So if we approach the hydrophobic surfaces together, then we expect hydrophobic attraction. Okay, so in the case of methyl-terminated SAMs, so we observed hydrophobic attraction and the van der Waals attraction. In the case of charged surfaces, so they are hydrophilic, so just a van der Waals interaction. But for the nonfouling SAMs, so this is a result of a protein adsorption and the adhesion. So if the surface is protein resistant or blood compatible, so we always observe repulsive force. So if we consider the origin of this repulsion, so between the surfaces, so we have only ions and water. If we consider the possibility of the electric double layer force, so the force should decay with the just decay length of the solution. But the decay length of this repulsion is at about 2 or 3 nanometer. But Debye length, so ion screening length is 0.7 nanometer. So very long range to compare with that length. So it's not an electric double layer force. So it's most likely water-induced force. So to prove this, so we performed such kind of experiment. So if we have a water-induced force, so if we add some alcohols, which disrupts the hydrogen bonding network of water molecules. So if it's a water-induced force, it may change by adding alcohols. So as expected, so we add ethanol. So this is a 0.3 mole to mole and just a 1-to-1 in a volume. Then we observed that this repulsion completely disappeared. So from this result, so near the bioinert sands, there is a kind of a water barrier which prevents the approach of cells or proteins. So we performed more systematic work. So we changed the number of ethylene glycol unit and terminal groups, and of course, depending on them, so the blood compatibility changes. But in this case, so if the surface is blood compatible, we always observe a repulsive force, water-induced repulsion. And if the surface is not blood compatible, we always observe the attraction. It's very clear. So far, 100% correlation. So I want to introduce one more interesting system. So this is oligoethylene glycol terminated SAMs, and number of ethylene glycol is 3, and the terminal group is a matrix group. So we made a monolayer on a gold and silver, and actually on gold and silver, the molecular packing densities are different. So on a gold surface, the density is a little bit lower compared with the silver supported SAMs. So macroscopic wettability, water contact angles are completely same. So we cannot distinguish. But the response of the proteins or cells are completely different. So gold supported SAMs shows excellent protein resistance, but silver supported SAMs is a protein adsorbing. And if we can compare the result of a cell adhesion, so the difference is very clear. So this is a Hubeck, and this is a result of the platelet. So completely different cell resistance depending on the substrate. And actually, so we try to observe the hydrogen bonding network of the interfacial water using the surface enhanced infrared adsorption spectroscopy. So this is based on total internal reflection IR, but we formed the gold film on the surface of the prism, and this single layer works as an antenna. Then, so we can generate a highly localized electromagnetic field near the surface. So using this electromagnetic field, we can selectively measure the IR spectra of the interfacial region. This is a collaboration with Morita Sensei in Osaka Electric and Communication University. So if you compare the OH stretching band of these SAMs, so depending on terminal groups, so completely different hydrogen bonding state. So this means so terminal groups really affect the hydrogen bonding network of the interfacial water. And we also measure the surface force between the SAMs. So on the gold surface, gold supported SAMs, so we observe the water-induced force. But the protein adsorbing silver supported SAM shows just a van der Waals interaction, no water barrier. So in this case, so we also observed that there is a water barrier or not. It's well correlated with blood compatibility. Actually, next question is what is the difference in molecular behavior at the interface? So we performed a computer simulation using a grand canonical Monte Carlo technique. And this is a model. So we constructed the cells in a simulation cell and put out many water molecules. And here is a interface. And so the packing density is based on XPS measurement. And we analyzed the density profile as a function of the distance from the surface. So this is a substrate, and this is a bulk water region. So green curve is a density profile of the water. And then we can compare the gold and silver. So in a gold film, in a gold supported film, so here is some water interface, and there is a primary hydration shell layer and the secondary hydration layer. And here it's constant bulk water. And some water molecules can penetrate into the cells. And there are several peaks. The position of the peaks corresponds to the oxygen atom of the ethylene glycol. So water molecules are trapped to the hydrogen bonding to the oligoethylene glycol. But in the case of silver supported cells, it's completely different. The molecules are densely packed, and such kind of monolayer don't allow the penetration of water into cells. So this is a main difference between the silver supported and the gold supported cells. And actually this water penetration was confirmed by the neutron reflectivity measurement. So what we expect is so there is an intermixing of water and PEG at the interface. And if we mix the water and PEG, so the dynamics of water around the polymer chain becomes very slow. And this interaction induces a clustering of water molecules around the polymer chain. And some computer simulation suggests such kind of clustered water induce repulsive force. Actually, we guessed such kind of water clustered water is a physical barrier for the protein resistance or cell resistance. And recently we performed the FM frequency modulation AFM, and this technique can visualize the surface structure with an atomic scale. So this is an AFM image of the mixed charge type blood compatible cells. So as illustrated, the molecules are periodically packed. Then, so not only the XY imaging, so we also can measure in the direction of Z. So this is a XZ or YZ cross-sectional view, and here it is the topmost of surface monolayers, and you can clearly see. So such kind of barrier is covering the cells. So this is a water barrier we discussed. So such kind of water barrier prevents the approach of the biomolecules or cells. So far we discussed about the artificial cells, artificial materials but how about biomolecules constituting our body? So proteins, saccharides, lipids, DNA. So this is a illustration. Now the distribution of macromolecules, biomolecules inside the cell. And this illustration is based on a real experimental result, real concentration. So what the molecules are really clouded. So we call this clouding condition. And but the biomolecules can react or bind to the target molecules accurately in such kind of situation. Very smart. So if we compare with our scales, so this is a photo of a swimming pool in Tokyo. It's really crowded. So if we lose our family in such a situation, so we cannot find out. So, but biomolecules can do. So we will discuss why. So 5 or 6 years ago, so we found a very exciting paper. This was published by Andrew White, Professor Andrew White. Now he's working in University of Rochester University in the United States. And he analyzed the composition of amino acid of protein surface. So protein surface is the stage of interaction, intermolecular interaction. And what he found is glutamic acid and the lysine is first and second abundant amino acid residues. And interestingly, so glutamic acid expressed in alphabet is E and lysine is a K. So this zwitterionic pair is frequently found on the protein surface. And based on this finding, so we also made such kind of zwitterionic peptide surface monolayers. So most abundant EK and DK, and a little bit different zwitterionic pairs, ER and DR. And we found such kind of monolayers are very bioinert, resistant and cell resistant. And if we compare the surface forces, so EK and DK surface monolayers have a water barrier, but ER, DR, so they have just a very thin water layer. That's why they cannot prevent the approach of proteins or cells. So protein molecules may have such kind of water barrier. And I try to visualize so how the situation is. Then, so this is a BSA albumin, so frequently used as a blocking agent in the biosensing. So if we emphasize the EK and the DK pairs, so it's uniformly distributed on the surface. So the other one is a molecular chaperone. So it recovers the native structure of proteins inside. And we found in the inner walls, EK and DK pairs are concentrated. So in the inside, in the cavity, so they reject the interaction with other protein molecules, which will be recovered. And the ferritin is also cage-type proteins, and it transports ions by accommodating the ion oxide inside. So it shows not so much EK and DK inside, but outside, a lot of EK and DK. So on the other hand, so we have investigated protein molecules which show the specific interaction with target molecules. For example, integrin, so this is a cell adhesion proteins. the density of the EK and DK is very low, and vitronectin and the fibronectin, so they provide GD site for the cell adhesion. So the population density is very low, and the EGG, and so this is responsible for our immune system. So EK and DK locate with avoiding the binding site. So if there is a EK and DK near the binding site, they reject everything with the water barrier. That's why they don't have EK and DK at the binding site. And the streptavidin. So this streptavidin and the biotin is the strongest non-covalent bond in the biological systems. So surprisingly, so this streptavidin has no EK and DK. So I guess, so we can guess, so this is protein preferred. So put the priority on strong binding, fast reaction. So we also found such kind of things. And actually, similar thing was found for the DNA cells. So this is a schematic image of the surface monolayers of DNA, and we changed. So most of them are complementary, but we modified. We just introduced one mismatch at the terminal groups of the helix. Then, so if we introduce the mismatch here, then so we covered the gold nanoparticles with these DNA molecules, and colloidal stability of the gold nanoparticles are really increased. On the other hand, so if we have no mismatch, the colloidal stability is very low. So we also observed the surface force, and we found a water barrier for the DNA with mismatch. And we just observed the hydrophobic attraction for the complementary DNA cells. So just by collecting together, so we can expect the biomolecules are using interfacial water smartly to enhance the accuracy of the molecular recognition. So in a summary, so we observed the physical barrier of water near the cells, and artificial molecules, and we found a water barrier to prevent and approach of cells or proteins. And for the biomolecules, so far, bioinert peptides and DNA cells, and they have also water barriers. Now we are working on saccharide-based surface monolayers. So we expect we will get a similar results. And actually, I talked very slow, and I will skip the data analysis part. So actually, I try to just overview. I try to find the hidden rule for the bioinertness, and we made a database of chemical structure and protein resistance, and then we performed the machine learning, and finally we succeeded in prediction of water contact angles and adsorption of proteins from the chemical structure of molecules constituting the monolayers. And actually, this approach is very useful, and recently we succeeded in a prediction of protein adsorption onto the more complex system, polymer brush systems. Yeah, and so finally, I would like to ask you several hints. So to be honest, all questions has not been solved. So most fundamental thing is why can water induce repulsion? So we don't have a detailed picture at the molecular level, and especially in this community, so many people are interested in exclusion zone water, easy water. There is some connection between them. So, and we are also interested in how hydrogen bonding states are modulated with the interaction with the surface. So actually, I want to review previous literatures, and I will define what remains unclear. So if we review the interaction in the water, so water-induced force sometimes oscillatory, so it's oscillating, and sometimes the interaction in the water is attraction. So the origin is a van der Waals attraction, hydrophobic interaction. So range is in the case of a van der Waals force, so angstrom to several nanometer. And the hydrophobic interaction is a range of several nanometer. And capillary evaporation and nanobubbles are also origin of the very ranged interaction. As for the repulsion, of course, so electronic double layer force, DLBO force, is one of the origins. And we can also have a possibility of a repulsive van der Waals force. And we can also observe the solvent-induced force, hydration force, sometimes osmotic force, and Lewis acid repulsion, or sometimes called as a hydrophilic interaction. But there are so many contradicting examples. So I just briefly introduce the oscillatory force. So there is a very clear conclusion. So this is a confining effect. So first, most famous one is the results by the Professor Israel Lativeri, and he measured the oscillation of forces by confining the water into the several nanometer. Then the periodicity of the oscillation is just the size of water molecules, 2.5 angstroms. It's a very beautiful oscillation. So if we confine the nanoparticles or rods or wires, so we can also observe the oscillating forces. It depends on shapes. And in the case of silica colloid, so we can observe the oscillation with the size of the silica colloids confined between two surfaces. But different from the previous example, attraction observed between hydrophobic surfaces, it's really a lot of discussions and contradicting results. For example, Israel Lativeri, so he's a godfather of surface force, and he just found that hydrophobic attraction with a range of several nanometer. But in AFM guides found a completely different range. So the range of attraction is up to 100 nanometer. And for the super hydrophobic surfaces, the range is just several hundreds of nanometer, from several nanometer to several hundreds of nanometer. It's it's a huge difference. But so recently, this problem was completely solved. So this was a effect of nanobubbles. So Professor Higashitani's group in the Kyoto University, so they measured the interaction between hydrophobic surfaces with normal water and degassed water. So if we use undegassed water, so we always observe a extremely long range attraction. But once the water is degassed, so just several nanometer, so same result found by the Professor Israel Latvia. So if we image the surface in the water, it's very clear. So such kind of structures come from the nanobubbles stably formed on the surface. And if once it's degassed, so nanobubbles stick on the surface completely disappeared. So the mystery of the hydrophobic attraction is completely solved. But how about hydration force observed for the blood compatible biomaterials? So let me simply explain what kind of water-induced force, especially hydrophilic or hydrophobic surfaces. So this is very simple system. So in the water, we introduce two surfaces. And if the surface likes water, so water is comfortable in a confined region. So in such a situation, so if we approach two surfaces together, so we observe a repulsion because the water wants to stay here. On the other hand, the hydrophobic cells, so they hate, so water molecules hate this region, just to go outside. Then, so the interaction will be attractive. So this is a very thermodynamical expression, but a lot of discussions about this. For example, so Professor Israel Latvia, so they stated, so hydration force cannot be repulsive. So if the water molecules attach on the surface, of course, water molecules have a dipole moment. It's kind of surfaces are approaching together. So if the water molecules cannot maintain such kind of repulsive regime, so they just optimize their positions. Then, so dipole-dipole interaction will be like this. So such kind of situation is more energetically favorable, and this interaction is must be attractive. On the other hand, so Professor Van Hoos, he predicted a lot of hydrophilic repulsion, and sometimes so it exceed the van der Waals, overcome the van der Waals attraction. So I think this is a repulsive force we observed in our surface force analysis, but still it's not clear. But recently, our members, Miss Elisa Mendoza, so she did a really great work. So she measured the repulsive forces for the two kinds of surface monolayers, the zwitterionic type and the non-ionic type. And she averaged so many force curves. And these force, so repulsive force can be fitted with a double exponential function. So two exponential function with a different decay lengths are necessary to fit data. And she extracted a long-range component and a short-range component and compared. Then, so of course, sulfobutane, so it has a two charged groups. So it really attracts water. They really attract water strongly. So in this case, the short-range repulsion is strong, very strong for the SB cells. On the other hand, so the strong interaction with water is not so strong for the EG3OH. In this case, short-range repulsion is weaker, but it shows a stronger long-range repulsion. So I think there are two components: the local hydration and also ordering effect of surface and monolayers. But just this is a still unknown, unsolved problem. And we also systematically measure the OH stretching band with a surface enhanced IR. And we found that depending on the terminal groups, so we observed completely different spectra. And actually it's with each spectra with 5 peaks. This is an assignment for the bulk water. So DAA means so 2 acceptors, lone pairs, and the donor. So hydrogen is occupied for the hydrogen bonding. And so we found, so the components of DA, so located at the 3,430 wave number. This component is very important for the protein resistance. So actually we are considering what does it mean. So this is a so-called intermediate water in the polymer science, but which we don't know what kind of hydrogen bonding state. So now we are trying to perform some theoretical calculation to solve this problem. So finally, I would like to summarize just 2 messages. So importance of water in the biomaterial science. So especially the biocompatibility, blood compatibilities in resistance or cell resistance, the interfacial water is very important. And latter part, so I described, so actually we don't know the detail about the formation of water barrier. And so in the future, we have to control the water behavior to realize the desired function of materials. So what kind of approaches are okay? We don't know. So we have to try. So, but finally, I'd like to introduce very important description. So this is a summary of my PhD thesis. I performed some computer simulation of the water solid interface. And my conclusion was, so the protein resistance of cells can hardly be ascribed to the protective coat of structured water. So water is not so important. Water is not the key. This is a conclusion of my PhD thesis. And actually, safely I got a PhD, but this conclusion is wrong. So finally, I would like to acknowledge the group members. So they are working on these topics, and they are trying to deny my PhD thesis. And I also thank my collaborators and financial support. So thank you very much. This is the end of my talk.
Thank you very much, Professor Hayashi. I'm sorry that we are actually on Zoom, so you cannot hear the applause, because I would really, really give you a big applause. Thank you so much. So
Thank you.
I just wanted, I had so many questions, but then when you gave this last few slides, when you said about this wave number 3430, so when I recalculated this band, it turns out to be one of the bands that I also thought is involved with this barrier. And
30, just a moment, which slide?
So that's one of the latest slides. I guess that one. So 3430. So that one. So this band, it is very, so we also observe this band. So when you recalculate into the wavelengths, it is 1457, and 1457 is almost always combined with another band, 1425. So if absorption goes up at 1425, for example, it will be down at 1457. So they're constantly in correlation. And what we know from our studies, 1457 is definitely, I call it Fizian salt water. And I really think it's van der Waals interaction. While 1425, we found as the functionality of this band is related to fibrillation of protein adsorption to the surface. So they're opposite. So this was, for me now, to see in someone else's work, fantastic. And I really appreciate. So I have other, of course, comments, but of course, audience should come first. I'm sorry. So we're open for questions. Whoever wants to ask question, you can raise hands, or we have one question in the chat box, or actually it is a comment. Is it a comment? Yeah. Oh, we have few. Okay, we have a from Pierre first.
Yes, I'm trying to lead.
Okay. Maybe we can ask Pierre to talk.
Okay, just a second. Hello, everybody. Can you see me? Can you hear me? No, not yet, because there is.
We can hear you.
Yes, I can hear you.
Okay, wonderful. Thank you, Professor Hayashi. This was very important, because I consider the molecular approach as one of the underlying, let's say, toolboxes in order to understand what's happening on a microscopic scale. And that's what you've described in technical terms, using this self-assembly layer in terms of an arbitrary technical system, and then convert this into biological structures is crucial, as many of the problems in let's say, chronic diseases are associated to this misunderstanding. The way you describe the interaction and the measurements, I think the toolbox you provided allows us to understand what can be done when we extend let's say, the toolbox toys that we are using, by including also electrodynamic properties. I'm saying this because that what you described reminds me a lot of the Nafion-induced easy layers that you mentioned at the very end, and also during the center of your talk. And I have the impression that these analogies cannot be neglected, because there are so many similarities. You have the PTFE backbone of Nafion that are similar to your, let's say, gold or silver surface structure. And then you have the side chains, and in Nafion you have the sulfuric acid side chains, which are quite similar. And they induce this kind of very peculiar behavior of, let's say, particle separation from the surface of the membrane towards, let's say, the bulk water that stretches into it. And you mentioned it, it's 100 nanometers long. And I wonder, if you extend your approach, and this brings me to the question, the actual questions, if you extend your approach using all the tools you have available, from atomic force microscopy to the Raman spectroscopy, and why not near-infrared spectroscopy, by applying electromagnetic field gradients of an oscillating field that couple to the coherent phase. I'm saying coherent phase because I assume that there is a biphasic nature of water, which is the bulk, and the coherent clustering, which is nicely described by Emilio De Giudice. And there was a paper in, I think it was during one of the conferences in Switzerland, in Basel, if I'm not mistaken. I should send you the paper, where together with Professor Vladimir Voyekov, they tried to describe this effect using quantum electrodynamics. And they came about the same conclusion as you said. You can induce hydrophobic and hydrophilic properties. It's in either way, you just shift the frequency. And in this case, it's a very elegant tool where you can modulate, not just to, let's say, enforce the theory, but also to extend the atomistic molecular approach by encompassing a broader picture. And I think there's a huge potential in there, because this would also enable us to understand why, for example, technical fields like mobile communications or extremely low frequency fields have a biological effect, because they obviously interfere with this kind of oscillating patterns. And that the underlying, let's say, baseline would be to understand how electrodynamics and electrostatics interact in order to induce this peculiar biomolecular surface interaction. And with that, I stop. Otherwise, it's getting too much into details.
Yeah, thank you for your comment. Yeah, actually, so as you wrote in a chat, so
yeah,
we have to mention the connection with the easy water.
yeah, there is a connection, definitely.
Yeah, and actually, I'm interested in a difference in the range. So in our case, so we just observed several nanometer. So the range of several nanometer. I mean repulsion. But in the case of easy zone found for Nafion surface, it's several tenths of micrometer. So clearly observed by the optical microscope. And for me, such kind of long range is unbelievable, and expect some completely different mechanism. For example, actually, I suspect such kind of effect. So the polymer chains of the Nafion is fluctuating at the surface by hydrating. And so such kind of if we have such kind of layer, so this layer looks like exclusion zone. So but I'm not sure. Actually,
I think. Sorry.
Yeah.
Go ahead. Please. I didn't want to interrupt you.
Yeah, but I want to investigate such kind of things with AFM. Then I want to measure the AFM surface force for Nafion system, and maybe I can contribute to this.
I'm sure you can contribute to this phenomenon, because if you use AFM and you let's say apply an electromagnetic field gradient onto, let's say, the sample that you have, I'm sure the arrangement will change. I'm not sure whether you're aware of Professor Gilbert Ling's association induction hypothesis, where he says protein activity is blocked once you have coherent fraction of water, and reactivated, so protein becomes functional again when you induce decoherence. So you can modulate protein activity by just using that. And I think in the same way as you showed in this particular slide that you just popped up on the screen, you can induce, let's say, the polymer chain.
Yes,
but an alteration in the folding pattern, which results in a change of, let's say, the exclusion zone thickness. If we put it bluntly in this way.
Yeah, thank you for your comment. So we can also apply the voltage between probe and the substrate. It's possible. Just low bias. to avoid water splitting.
But no. We're talking not about strong field gradients anyway. Yeah. Now, this is fantastic. If you can do that, this would be groundbreaking stuff.
Mm.
Thank you for your positive comment. Yeah.
I would be also interested to see that result. Professor Voyakov, you have a question, I see.
Okay, thank you very much. Professor Hayashi, brilliant lecture, of course. And I would like, after Pierre, to return to the problem of easy water. You used as a substrate for all your monolayers silver or aurum or gold, as I understand. Yes. But it was shown by Franco Musumeci together with Jerry Pollock that near gold there is practically no easy water. But when they used as a metal zinc, there was about 100 micron easy water. Of course, in your case, you already applied to this substrate the molecules, the macromolecules. That may, of course, change the situation. But in any case, the question of what is the nature of substrate may be also very interesting. If you use not the inert metal, like silver, gold, or platinum, near platinum, they didn't see practically no easy water appeared. But if you use some active metal, probably situation will be different, but I don't know how it will affect the properties of the monolayer. So that's my remark probably, or even question. And thank you very much. It was really very, very interesting.
Thank you.
Thank you for your comment. Yeah.
Yes, this exclusion zone phenomena obviously has many questions. I remember that I, for example, found peculiar that on contact lenses, some of the contact lenses, for example, exclusion zone would form on one part, on concave or concave surface, but not on the other one. So the same material, just the curvature is different. So that was awesome. Professor Harada has a question. Could you please unmute?
Okay, thank you. I'm very fascinated with your talk. And I was also very surprised to see that the long-range repulsion is caused by the nanobubbles. Attraction is caused by nanobubbles. Yeah. So, but is it normal for nanobubbles to be on the surface without degassing? And what is its quantity, like the density per area?
So we actually so your question is
so is it normal? So actually now the people were discussing the presence or the stabilization of nanobubbles, but you said that it's always in liquid water. It seems like without degassing. So with degassing, there's no nanobubbles in the bottom, but so above we can see the nanobubbles there. But is it always staying there?
I think it's if without the degassing. So to be honest, we had a problem in we had a trouble of the formation of bubbles for the biosensing or surface force measurement. Especially when we study the hydrophobic surfaces. So yeah, for example, if we measure the protein adsorption onto the hydrophobic SAMs, so we sometimes get we detect the bubbles, not proteins. So so yeah, actually, so that degassing is really helps for the biosensing.
Okay, so one question is how much is the density you are expecting? Density of the nanobubbles?
Depends on the concentration of gas dissolved in a solution and the hydrophobicity of the surface, I think.
So they are okay. They are okay. Aggregating on the surface, the hydrophobic surface.
Yes.
Okay. Okay. Thank you very much.
We had a very interesting comment from Professor Takeuchi. Would you like to share? Thank you, Professor Harada. Takeuchi-sensei, are you there? Okay, Takeuchi-sensei perhaps left, but there was very interesting comment in the chat box.
Yes. Please.
I actually always expect from Takeuchi. Takeuchi-sensei is here. I always expect that Takeuchi-sensei is going to provide explanation of
I am doing
structure.
my experiment.
Sorry. I wanted this is very interesting comment. So maybe people would like to know
because
more about this.
When you discuss about the OH vibration band at around 360.
Yes. You mentioned
to 81 wave number to 1080 wave number region. The Fermi resonance between OH vibration and the second overtone of water bending mode is around 3200 wave numbers.
Yes, it's true. Yeah. So because of the Fermi resonance, so we cannot discuss in detail. But along with this, we also measure completely same measurement for the deuterated water, D2O. So we always compare the results. Yeah.
Okay.
Yeah. Thank you for your comment. Yeah.
Thank you.
That is very valuable thing to know. I will take this comment very close in my future experiments. Professor Tenkwa, you have a question. Could you please unmute?
Thank you. I was just adding. I was going to add something to the discussion about nanobubbles. We have found in our experiments that the presence of nanobubbles increased the solvation shell in the water. So obviously, what you showed in the infrared spectra slide, when the long range interactions are related to less hydrogen bonded water, obviously, this is a very nice consistency with what we found, the presence of solvation shells surrounding the bubbles. So exactly. This is the slide. Yeah. So I think something. Yeah. What we can do is really to repeat the experiment in infrared and just see the presence of because we have identified the specific absorbent band of water solvation shell, also the dehydration band and hydration band, as well as Jelena mentioned, the protein hydration band. So those few bands might help understanding what's happening in a media with bubbles. Yeah.
Actually, yeah. Thank you for your comment. To be honest, I don't know. This is effect of bubbles or hydrophobic surface. But so if we check the spectra very carefully, so we have a very sharp peaks for the isolated OH. So water, OH bond without the hydrogen bonding. We don't know this is against the bubbles or hydrophobic surface. And if the actually bubble and the hydrophobic surface, so they are hydrophobic, and so interfacial water try to maximize the hydrogen bonding with their surroundings. So this lowers the vibrational energy. So you can see the lowering of I think this is what we found. But yeah, I think your comment is right. So
yeah, you might have both phenomena at the same place, as you mentioned in your talk. So
I see.
Yeah.
Okay.
Thank you for your comment. Yeah.
Yes, thank you.
Thank you. We also had in a chat box a comment from, I'm not sure if I will read properly, Shogo Shigeoka-san. Would you like to comment? So the question was, can these SAMs be used to test chemical properties of terminal groups? And if so, how does the bonds between S and the AgAuPT layer alter the actual electrochemical potential of various terminal groups? So what's the influence between what's the actual influence of this layer of metal on the electrochemical potential of terminal groups?
What are the, just a moment. How does, so first question is, yeah, so interaction between sulfur and against the silver and the gold. So they are noble metals. Yeah, actually, the surface and monolayer alters the work function of the metal surface. So I think the electrochemical potential may change. And in a case of, so I mean, so I think the nature of the chemical bonds are very similar for these metals. But I'm not sure the change in the work function, because work function depending on molecular packing density. Maybe I think there are several works using the Kelvin probe microscope, and so when so it's question 2, when organizing your library and predicted deep learning algorithm, at least hidden. We don't have such kind of large scale database for the proteins. We just now we are performing the statistical analysis of the just population of zwitterionic EK and DK. So we don't have a large scale data set for the deep learning at the moment. But in future, yeah, we will do that. Is it did I answer your question? And
one more comment was about easy.
yes, that's all. So actually, we don't have near infrared spectroscopy. Maybe this will be the collaboration with Yelena.
Yes, I hope so. I certainly hope so. I really like your work. Okay, any more questions? We have some few more minutes, like 2 or 3 minutes, if someone has some short question. Otherwise, I will probably ask something. We have. Is it possible to see the? Oh, sorry, not really. Yes. Of course, we will have this video recorded at and put on our YouTube channel, so this can be viewed many times, as I will do certainly.
And it might be translated.
Yes. We will probably
in Japanese.
I really liked now when I was looking at my comments, I really liked how you said that based on this database that you created about the proteins that you found, that there is no correlation between batability and protein resistance. This is a really interesting thing. because I always, wettability is a phenomenon that for me was explained by Professor Takeuchi and his works, and basically by the ratio of, let's say, cohesive and adhesive forces. Actually, based on the ratio of free water molecules and bonded water molecules. So, and those bands, for example, in our near infrared have nothing to do with the bands that we see are associated, for example, with something like protein adhesion or this, as I said, bulk water, PCSO water, and so on. So that was really interesting. So Pierre has some recommendation, okay, for the origin and the special role of coherent water in living systems. Thank you, Pierre.
Thank you.
Yes, that is one more topic about which we should learn more. Coherent.
Yes, I will check it out. Thanks.
Yes, Professor actually Tomohiro Hayashi had a very nice comment about Pierre's lecture and coherence. For me, it was very inspiring. Okay, I'm giving a chance for one short question once more. And if there is no, that's 3, 2, 1. Discussion is now closed. I would like to thank you again to all of you. There will be now a short poll that you can answer our questions. So just a quick question: what would you like to? Did you like the webinar? Do you have some comments? Do you? What would you like to see in the future on our webinars? And of course, if you have more questions about this webinar, you can also put it in the comments on the YouTube channel when it is uploaded. We will send tomorrow basically the link for this for the webinar, so Professor Hayashi can also maybe take a look and respond. Just one more thing, just one more notice. So we will organize the next webinar in April to basically celebrate 1 year of our existence as an Aquaphotomics research department, and we're going to give something like a review of our works, our published papers, what we did, and what we achieved, and maybe we will definitely talk about some collaborations and what we're going to do in future. I would like once more to thank to Hayashi Sensei. Thank you very much. This was very, very inspiring talk. I really hope that we may join you to work with near infrared on this. It would be beautiful.
Thank you. Thank you for your arrangement. And thank you all for the comments. So if you have any questions, just send a question to Yelena and me, then we will answer.
Yes, you can always, I can forward emails to Hayashi Sensei. So we have just comments from Tiana. Thank you for the interesting lecture. Okay, well, with this, I want to thank you everyone for joining us today. Thank you, Professor Hayashi, and see you again in 1 month, hopefully. Okay, so bye-bye, Europe.
Thank you, Yelena.
Good night.
Good
night, Japan. Good night. And Serbia do Tokyo. Goodbye.